development program. So first of all, I'd like to just give you a little overview of where this particular program fits within the HEAL initiative. So for those of you who aren't familiar with the HEAL initiative, HEAL stands for Helping to End Addiction Long Term. And it's actually a really broad, multiple institute within the NIH type of program, and it's got multiple research objectives. Our particular program, the Pain Therapeutics Development Program, falls within the objective to enhance pain management. Some of the things that are part of that particular research objective are to understand the biological underpinnings of chronic pain. And the one, of course, that this program fits in is to accelerate the discovery and preclinical development of non-addictive pain treatments. Other aspects of the pain management objective are to advance new non-addictive pain treatments through the clinical pipeline. And another one is to inform best practices for effective pain management while minimizing the risk of addiction. You could, if you're interested in just knowing more about the HEAL initiative, you can find it online and read about the research plan at www.nih.gov HEAL initiative. Or you can check out the JAMA paper that was written in 2018 that pretty much outlined the overview of what the program's objectives are and exactly how it's set up. So within the sub-objective of developing therapeutics, the HEAL initiative actually has multiple analgesic development programs that cover pretty much the entire early drug development spectrum. So in this particular slide, you see the chevron showing discovery, development, clinical testing, and implementation. And several of our programs cover the discovery, development, and early clinical space. So the first one, the one that's earliest, is a planning grant. That one is RFA NS 21-029, and that covers activities such as planning the studies that you want to do, getting a team together, understanding a little bit more about the preliminary biology and mechanism behind the biological target, things like that. And I'll be talking a little bit more about that in a few minutes. And the follow-up funding opportunity covers assay development, screening, initial hit characterization, and biomarkers. And that is, I'm just going to refer to that as the team research grant. It's basically the grant that you can get into after you've completed the planning activities in the one prior to that. And that is RFA NS 21015. And then the Pain Therapeutics Development Program covers it does overlap to a certain extent for things like screening and initial hit characterization from the team research grant, but it also covers later activities such as hit to lead characterization, lead characterization itself, IND enabling studies, and phase 1 clinical trials. And the funding opportunity for that one is RFA NS 21010. And then along with that, some of the NCATS, which is another institute within the NIH, is also part of this whole general HEAL therapeutics initiative. And they provide translational assays, interesting ones such as organ on a chip. And then we also have a program called the Preclinical Screening Program that does preclinical therapeutic profiling of a particular asset. We'll give you, and that's on the efficacy side. So we'll give you efficacy profile in different models of pain. And it will also provide just basic pharmacokinetics on the asset that is being submitted. And then finally, as you can see, the program that goes furthest to clinical testing is the Pain Therapeutics Development Program, and it goes to phase 1. And so phase 2 could be conducted if you apply to EPPIC-Net, which does conduct phase 2 clinical trials. But again, every single one of these are grant or contract opportunities that you have to apply for. There are 10 participating NIH institutes in the analgesic development programs, and they include NINDS, NIAAA, NIDA, NCCIH, NIAMS, NICHD, NIDCR, NIDDK, NEI, and NHLBI. So now just to focus for just one slide on the planning grant and the team translational grant. So the planning grant I was referring to in the previous slide it uses a mechanism called an R61. It's 2 years in length. It allows direct costs of $500,000 per year, and that includes consortium or subaward costs. And as I was mentioning, it's set up so that you can build your multidisciplinary team during that point, and you can obtain preliminary data that would be helpful in showing the robustness of your biological rationale for some of these more advanced funding opportunities. And it also directly provides the activities and information you need to apply to the U19 team management grant. And the entry criteria for that are to have a multidisciplinary team and showing evidence that the team has expertise and can work together. And another criteria is that you now have a biological rationale that addresses the feasibility of your therapeutic idea. And therefore it requires preliminary data or at least literature-based evidence. And some of that preliminary data, of course, can come from the R61 if you want to follow these grants in order. This U19 is up to 5 years if all milestones are met each year. And it allows direct costs of $1.5 million per year, again, including consortium and subaward costs. There is no opportunity for renewal of either the R61 or U19. Okay, now I just want to spend a little time also on the preclinical screening program for pain. That program is unlike the other two I just talked about. This program's purely based on a contract. And so this particular contract allows people to, apply to get their assets tested. So as these therapeutic assets could be biologics, they could be small molecules, they could be natural products or devices. And once they are accepted, asset testing is accepted for the PSPP program, then under NINDS direction, rigorous preclinical evaluation of these assets is conducted in a suite of assays that are performed by a contractor. That contractor is Psychogenics, which probably some of you are familiar with. And these studies are conducted in a blinded fashion and on a confidential basis at no cost to the participants. So this is a free resource to people who have their therapeutic assets accepted. In addition, the PSPP staff interacts with the participant to plan testing, review test results, and discuss future studies. And another important thing to point out about this program is the participants' intellectual property and confidentiality are absolutely protected. One goal for this is for therapeutic assets that are tested in this program and profiled to move perhaps forward towards clinical trials, or at least move forward into later stages of development, or to be perhaps partnered, licensed into a company. PSPP is open to academic, industry, and government asset owners worldwide. The program, one thing I didn't mention for the first one, the program director for this one is Smriti Iyengar, and the program director for the R61 and the U19 that I just talked about is Julia Bachman, and she's on this panel and can address questions about her programs during the Q&A session. So now to spend a little bit of time on the HEAL Therapeutics Development Program. This program really is very similar to a virtual pharma model because it involves virtual cross-functional teams, and it features a grant and contracts. So it combines the two types of resources. When one of these the grant is the cornerstone of this, just like the R61 and U19. But once a person's been awarded one of these grants, they form a lead development or lead product team. And the person in charge of it is the principal investigator. And the team also includes consultants that have quite a bit of industry experience and have expertise in different areas, and NIH staff. So the grant again is RFA NS 21010. And one of the things that the slide shows in the green box is that the PI is always responsible for pretty much all of the biology that goes into the project. So they're responsible for bioactivity assays, biomarkers, and efficacy studies, to name a few. But what they can do is they can take advantage of the contracts that NIH offers as part of this program. So these contract resources are tailored for each project, and the awardee, the grant awardee, can choose which NIH contracts that they want to use, because each project is different and each has different needs. And NIH consultants are assigned to every project. So that's one thing that every project gets, but they're tailored to the project based on the expertise needed for the project at the particular stage the project is in. And then one important thing is the PI's team's intellectual property is always retained by the PI's institution. And just to go over these contracts that we offer a medicinal chemistry contract, and the CRO there is Curia. They used to be AMRI. We offer a PK and toxicology contract, and the two contractors there are SRI and ITRI. We offer a data management contract with Collaborative Drug Discovery, or CDD. Every project we have uses this particular contract just to keep data in a secure vault. We have contracts for manufacturing and formulation, MRI Global and Curia. And then we also have two CROs, two vendors that conduct phase 1 clinical trials, and that's Duke University and PPD. So a little bit more about the grant itself. So the goal of this particular grant, RFA-NS 21010, is to accelerate the development of novel, non-opioid, non-addictive analgesics. And that, of course, is the goal of all of our grants. Some specific features of this particular grant are that it's a phased, milestone-driven cooperative agreement grant. It supports hit-to-lead activities, also supports lead optimization and characterization. It supports IND enabling studies and phase 1 trials, and as I mentioned, provides access to expert scientific consultants. Some of the end goals and milestones for this funding opportunity are to identify and fully characterize a lead candidate, to identify target engagement biomarkers or a biomarker if possible. We understand that this is not always possible, but this is something that we do strive for. An end goal is to seek partnerships. Another one is to complete your IND or BLA enabling studies, and then to file the IND or BLA. And keep in mind that I'm actually mentioning BLA along with IND because this program also takes biologics as well as small molecules. And then another end goal is to complete phase 1 clinical trials and be ready for a phase 2 clinical trial. So another interesting thing about this funding opportunity is that it does have quite a bit of flexibility. So we understand that people will have projects that are in various stages of drug discovery or early drug development. So they can enter at multiple stages of development. But one thing that every project must do is they all must begin with the preparatory UG3 phase. So you can see in this slide that the entry phase can range from the very early exploratory all the way into pre-development or IND enabling. We define discovery in that at least the entry criteria for hit selection are met, and we'll go over entry criteria in a few minutes. And we define development as entry criteria have been completed for IND enabling. The duration of the UG3, UH3 two-phased grant is 5 years maximum in combination. And usually the way things are set up is that the UG3 phase is for 2 years maximum. We have people come in with just using 1 year. And the UH3 phase is usually around 3 years. And one thing to stress is that your application must include both plans for both the UG3 and UH3 activities in the application and in your budgets. And then it is also important to lay out your path to clinical testing. Even if you just do it in a fairly non-detailed fashion, it's important for reviewers to understand that you know the entire path that you intend to carry out with your project. All right, and so who is eligible for this particular grant? Academic institutions are eligible, small businesses, and foreign institutions and components are also eligible. A note about applications from foreign organizations is that reviewers will assess whether the project presents special opportunities for furthering research programs through the use of unusual talent, resources, populations, or environmental conditions that exist in other countries and either are not readily available in the United States or augment US resources. But a justification has to be made when an application does come in from a foreign institution. Okay, now to just go over some of the entry criteria for RFA-NS 21010. So for if you are coming in the discovery phase, if you think your project is early, in other words, let's say that you have a set of potential hits, whether they're biologic or small molecule, and your assays are set up and ready to go and working in your lab. But now you want to actually do some structure activity relationship work, then you would be in discovery. So entry criteria to get into discovery are a rigorous biological rationale for the intended approach, a promising small molecule or biologic starting point for optimization, and scientifically sound assays to optimize and test the agent. If you think you're in the development stage and you want to enter there, then the entry criteria are to have identified a candidate therapeutic. So that's non-negotiable, that has to be there. And then also, if you're in the development stage, your application has to include a strong data package that links modulation of the therapeutic target by the candidate therapeutic to disease modification. Another entry criteria is that biological activity and ADMET properties of the candidate therapeutic are appropriate for the intended use. ADMET means absorption, distribution, metabolism, excretion, and toxicity. Okay. So since we just got through talking about entry criteria, I should just mention something about basis for application withdrawal. And applications can be withdrawn for proposing non-responsive activities. Here are actually all of the non-responsive activities. If your purpose of your grant is to screen for initial hit identification, that is non-responsive. We understand that you may continue screening while you're doing structure activity relationship work, that's fine. But if you're trying to find your initial hit, then you belong probably in the U19 we were just talking about. Another non-responsive activity is basic research and studies of disease mechanism. Animal model development is non-responsive. Development of diagnostic and diagnostic devices are not responsive. Clinical studies that are beyond phase 1 testing are non-responsive. And then here's another important one. Projects that propose therapeutics that are for opioid sparing or projects that are still targeting the new opioid receptor are non-responsive because with HEAL, the whole point is non-addictive pain medications. So to give you an idea of what some of the activities are in the two phases of the UG3-UH3, I've listed some in this slide. So in the preparatory UG3 phase, typical activities are optimization of the hits that you have, again, biologic or small molecule, using potency and efficacy screens. You could also be doing preliminary efficacy and toxicology testing in appropriate animal models for pain. Another common activity is characterization and testing for ADME, absorption, distribution, metabolism, and excretion. And usually these are in vitro tests at this particular point. And then also initial development of pharmacodynamic or target engagement biomarkers associated with the therapeutic target or the pathway. Then if you move to the execution or UH3 phase, activities typical for that phase are to continue some optimization activities if needed. And then also to begin conducting pharmacokinetics testing, biodistribution, gene expression, tumorigenicity, and immunogenicity testing. Again, for biologics, cell bank development and testing would be appropriate. And then for either biologics or small molecules, formulation and manufacturing, so that you have material for toxicology and human clinical studies are part of the UH3 phase. IND enabling characterization of the clinical candidate would be done in this phase. Non-GLP and GLP tox studies the same way. And IND submission and phase 1 clinical testing is also appropriate for this phase. In this phase, I should mention that you do not absolutely have to conduct phase 1 clinical testing as an activity within this funding opportunity announcement. That's up to you, but that is available. And we do have a lot of our applicants do that. Okay, so then you might be asking yourselves, how is it that if you have to apply for both the UG3 and UH3 phase in the initial application, how do you move from that UG3 to UH3 phase? So the way that happens is at the end of the UG3 phase, NIH program staff and leadership will determine if the project will advance to the UH3 phase. So this is an administrative decision that doesn't go back out to peer review. At the end of the UG3 phase, NIH program staff and leadership make these decisions, but I should mention, not only at the end of the UG3 phase, but also annually, NIH program staff and leadership conduct an annual administrative review. Reviews are based on things like successful achievement of milestones, the overall feasibility of project advancement, including biomarker work and considering data that may not have been captured in the milestones proposed in the application. Because you do have to remember that every time we're looking at project advancement decisions and milestones, we're looking at the potential for the project to continue on and get to phase 1. And if it looks like there's no place to go anymore, that's when there could be a decision about not continuing the project. Reviews are also based on the competitive landscape for the disease indication and drug target. They can be based on program priorities and availability of funds. So I've mentioned milestones a couple of times. So I want to go into that a little bit more to perhaps help everybody understand what we mean when we talk about milestones. So first of all, they're a very important part of the administrative decision to transition to UH3. And actually, they're also very important in each of those annual milestone discussions. Because to be clear, if you do not meet milestones at the end of a year, let's say, especially in UH3, you could also have the grant terminated. So grant termination could essentially occur at any year of the project. Then milestones must be included in the grant application. That's another thing to remember. That would be missing information if they were not included. What are they used for? They're used to measure success in achieving each of the research plan's key objectives. One or more milestones should be used for each of your key objectives. For each milestone, it's important to provide details on the methods, the assumptions, experimental designs, and data analysis plans. You should specify the quantitative criteria for measuring success and the related rationale behind that criteria. Quantitative criteria should be robust and they should be consistent with the state of the art in the field. That's one of the things reviewers do look at. The quantitative criteria for success in the milestones will be used for making go, no-go decisions, and this should be specified. So we usually tell people when they're coming up with their milestones that they think of all of them as go, no-go decision-making milestones. They shouldn't be just an activity that's listed. They should actually have some relationship to results that are needed to convince the team to move forward and that it's feasible to move forward. It's also important to specify the timeline for each milestone. There should be at least one milestone, of course, each year. And milestones, this is important to remember too, milestones will be further refined after the award with the NIH staff and the lead development team that you have been assigned to and that you will now be the chair of. So spend a little bit of time on intellectual property expectations. Here's just a little overview of some of the assumptions that we have. Inventorship is the same as in any patent, so it's determined by US patent law. The NIH Therapeutics Development Program has absolutely no stake in your intellectual property. We want that to completely belong to you because we want you to be able to eventually commercialize your therapeutic and get it to patients. Prior to grant award, your institution must have upfront intellectual property agreements in place with all potential inventors. And when we say potential inventors, we're talking about, for instance, the consultants that will be assigned to your lead development team. They have to have an upfront IP agreement in place with your institution, assigning intellectual property to you. And the CROs are the same way. So these agreements must address who will hold title to the intellectual property on new chemical matter or biologics or use patents, and also should address royalty arrangements. And that those things, royalty arrangements can be agreed upon between your institution and the consultants or the CROs, or they can be completely assigned to you, but that would be up to your institution. IP agreement should aim for unencumbered intellectual property consistent with our goal to create a licensable product. So here are some things that it would be good just to make sure that your application includes. So your application should have separate budgets for each year of the grant. For example, a grant of 5 years should have 5 separate sets of budget sheets plus the cumulative budget. Your grant should, in your grant, it should be clearly indicated with a table which activities will be conducted by you or the program director and associated personnel funded by the UG3 UH3 award, and which activities will be conducted by NIH contract research organizations and or consultants. So it helps us, just I can't say this enough, it helps us greatly to have that table that specifically outlines activities and who's going to be conducting them, you and your institution or NIH contract organizations. That helps us plan. It's also important to include a table of proposed activities that provides the following information. Activity itself, whether it's an optimization effort, assays, PK, et cetera, throughput, how many samples per month do you expect? Who's going to be doing these activities? Again, you as the PI, maybe a sub-awardee of your grant, NIH contractor, but we need to know a table of responsibilities. And then we also need to know what your early advancement criteria are to move from one stage to the next in your project. Your grant should also include a target product profile table. And this this target product profile is more on the clinical end. It's once you have a clinical candidate, and once you actually, let's say once you have an approved drug, what do you want it to look like? What is the acceptable situation and what is the ideal situation? So in this table, you would include things like what is the patient population you are shooting for in the ideal or the acceptable situation? What is the route of administration you want in the clinic? So things like that would have to be addressed there. And you're back to what your application needs to include. It should have a biomarker component in that's addressed at least. It should also address intellectual property and the other attachments section of your application. And then another little hint is if you are asking NIH to conduct the clinical trial, the phase 1 trial, using our contracts, then you should mark application clinical trial equals no. If you're going to do it yourself, most people mark delayed onset clinical trial. So another reminder here is that when this particular FOA was first published, we had a statement in there saying that people were going to have to do cost sharing, a cost matching requirement. Instruction was provided and had to be in letters of support. But for now, we removed that cost matching requirement. So that's no longer important anymore. So whether you're a small business, especially if you're a small business, you don't have to worry about that. Okay, some of the things that are also important here when you're applying is please really read the funding opportunity announcement very carefully. There's a lot in there, but there's a lot of really clear instructions which should help you. And another really important thing we can't stress enough is do discuss your proposal with us. So feel free to contact the program directors that are on the funding opportunity announcement, or you can go to our website and you'll see who the program directors are for this particular funding opportunity. It's important to stick to your page limit that's outlined in the instructions. And in the scientific premise, it's important that you explicitly discuss the quality of the data presented in prior publications in a detailed manner. And you also need to address rigor in your application. So you should detail the controls being used for each type of experiment and appropriately highlight potential confounds like surgery exposure, genotype, culture to culture variability, and human placebo effects. It's also important to include details within the experimental design about the reduction of potential bias, including things like blinding, randomization, and inclusion-exclusion criteria. And then you should also describe the source of the data on which the sample size estimation was based, and you should outline your power analysis. A few additional things, you should complete your required registrations for the application at least 6 to 8 weeks in advance of the receipt dates that are listed on the application. You should consider submitting your application early. It gives you a chance to react to issues that might result from your application being withdrawn. So just make sure you plan ahead a little bit. Discuss your budget with NIH staff if you do have questions. And another important thing is if you are applying to this program and any proposed budget year exceeds $500,000 in direct costs, then you need permission from NIH to submit. So this request for review basically must be made 6 to 8 weeks prior to the submission date on the application, on the FOA. And then another important thing is do talk with your tech transfer and business development group, particularly about the intellectual property section. And then you also will talk to them because you need to plan for funding patents and licensing activities as well. And then things that you should keep in mind that you don't want to do is please don't plan for this to be the sole funding for your lab, because as I mentioned a couple times, this is a milestone-driven program and it can end abruptly. And do not forget to address the use of NIH contract resources. You know, and, you know, really think through, you have the choice of being able to do your own subcontracting and putting that into your application budget, or you can use our contracts, but think strategically about how you want to, you can mix and match that too. You can use some NIH contract resources, do some subcontracting on your own. But one thing, a couple things to keep in mind: do not plan for NIH contracts to do your disease biology. That will be up to you. We do not have any contracts that address disease biology. And the only one that comes close is applying to the PSPP program. And again, they don't take everybody for that. And that is basically to profile on an in vivo basis your therapeutic asset in a number of pain models, or whichever ones you suggest. Please do not include NIH contracts in the application budget, because those are free to you, so you don't need to put them in the budget. And because of that, you should not reach out to planned NIH contractors for letters of support, because we are the sponsors for those NIH contractors, so nothing would have to come through you for that. Okay. And so then this is the time to start the questions, and we can have a little discussion on anything you want to talk about regarding these funding opportunities. I just wanted to point out that I'm the program lead for this particular funding opportunity, RFANS 21010. The health program specialist for the opportunity is Carolyn Bondar, who introduced this particular webinar. The Blueprint Neurotherapeutic Program Director, who also is in charge of all, this particular program, is Charles Sywin. And our program coordinator is Wakanda Medley. And in terms of remaining receipt dates, we have a receipt, the next one's February 15th, 2022. And we have receipt dates going out through October 17th, 2023. So that's it. And please feel free to ask questions.
Thanks, Marianne. We have one question so far. For UG3, UH3, did you say it's 500,000 K per year?
Yes, yes. In any particular year, if you meet that direct cost of $500,000, then you need to apply to get your application reviewed. So you need to contact us. And that's 6 to 8 weeks ahead of the receipt date.
Okay, next question. How does one conduct a phase 1 study at 500K?
So that's why this isn't saying that there's a $500,000 cap on any one year. It's just that you have to get permission to be reviewed if you go above $500,000. So, I mean, obviously one way to do this, of course, is if you don't want to bother with that, you know, and you can use healthy human subjects for your phase 1A. You could go through us, and then you don't have to worry about it at all. Or if you need to do it yourself, you can go obviously above 500,000, just because it's reviewed just to make sure that it's something that's feasible within budgets that are allowed within HEAL. So, you know, it's usually if you provide it, we actually provide you with a list of things you would have to address for that review. And we can actually do that. So it's not so difficult to do that. And actually, you know, this, I should say for HEAL, because it's an RFA, forget what I said about the 500,000. I made a mistake. So forget about the 500,000 direct costs for that one, because actually, I don't think that applies. So don't worry about it. we'll wait a few more minutes for any other questions.
Looks like that may be about it, Marianne.
Yeah, that's what I was thinking. And so everyone, if you think of other questions, feel free to go to the website. The recording will be available. And Rakonda, will the slides be available also?
Yes, they will.
Yeah. All right. And also, if you have any questions about your specific projects or applications, you feel free to contact us as well. We're always happy to talk to you.